Welcome to today's Tissue Gnostics webinar. My name is Florian Gruber, and later on Christopher Kremslehner will take over. We are from the Department of Dermatology of the Medical University Vienna in Austria. And we are the Christian Doppler Laboratory on Skin Multimodal Imaging of Aging and Senescence, SKINMAGINE. And what we do is multimodal imaging of cellular senescence. And we will show you today how we do that using tissue cytometry and other technologies. So what is senescence? How does the senescence process of cells contribute to the aging of skin and age-related changes that we observe in the skin? So our lab investigates skin aging, a process that leaves skin less functional, with lower ability to regenerate after disturbances or after damage, and also is aesthetically less appealing. We all feel that. There are several characteristic features of aging at the cellular level called hallmarks of aging. And these define the aging phenotype, and these include reduced genomic stability and alterations to telomeres and chromosome characteristics, but also dysregulated metabolic functions, and generally a loss of the cellular quality control. And in the last years, it became evident that so-called senescent cells are not just another hallmark, but that they are rather at the center of several of these processes. And these senescent cells, they usually show a kind of a damaged phenotype. So cells, when they have a damage that needs to be repaired, they go into a state called cellular senescence. And what their main feature is, they don't proliferate anymore, but also they don't die. And if they hang around for some time, they produce they start to produce a cocktail of inflammatory cytokines, proteases, MMPs, and other signaling compounds that together can damage the tissue if these cells reside in the tissue for a longer time, and they also elicit this inflammatory phenotype in aged tissues, which is often observed in aged tissues. So as the markers that define senescent cells are not really unique for the senescence phenotype, for example, cell cycle arrest, it became consensus that a combination of several markers must be evident to define a cell as being senescent, especially when we are talking about in situ characterization of senescent cells. We have just recently published in Cell the guidelines which experimentation would be ideal for defining senescent cells in vivo. Features that can be combined then as markers are, for example, classical cell cycle arrest markers or markers of DNA damage or DNA damage repair, rather. And additionally, markers for the erosion of the nuclear envelope, for example, staining for lamin B1. Then markers that highlight the inflammatory phenotype of the senescent cells, the so-called SASP, the senescence-associated secretory phenotype, can be added for the detection, like expression of proteases or activation of alamines or cytokine production. And additionally, and it's also something we are investigating specifically, markers for oxidative damage and its consequences. For example, protein oxidation or lipid oxidation that causes protein oxidation, and markers also for metabolic alterations would be applicable to define senescent cells together with the growth arrest markers and the SASP. And we study the combination of such markers in the skin. However, the skin is a quite complex tissue. And because specifically in the epidermis, so the outermost layers of the skin, we do not only have aging and senescence parameters that we observe, but overlaying that is this a constant say regeneration and renewal of the epithelial cell of the keratinocytes. We have here a basal layer of the cells, and there are also stem cells and precursors for the epithelial cells, for the keratinocytes of the epidermis. And these precursors show a lot of cell divisions to replenish both the basal layer and of the epidermis, and they also have asymmetric cell divisions, which give rise to daughters, which then undergo the so-called terminal differentiation process. And this process transforms the cells in the end to these dead corneocytes, which are shed off. But they form the horny layer, which is such an important part of the skin's barrier to the outside. During the differentiation, however, there is a massive changes in the cell content, in the biochemistry, in the activity, metabolism, lipid content of these cells. And thus it is difficult to study additionally in this scenario the aging of cells in situ. So what we are doing in the Christian Doppler laboratory SKINMAGINE is to investigate the development and the consequences of cellular senescence in skin, how it is promoted by extrinsic factors like pollutants, and most important for skin also ultraviolet radiation. And besides of classical biochemical and cell biological methods, we use so-called multimodal analytical imaging approach, in which we combine novel modalities like, for example, mass spectrometry imaging with spectroscopy methods and somatic activity imaging and classical immunofluorescence staining, of course, which we then investigate using tissue cytometry. And thereby we want, of course, to gain a kind of holistic insight on the senescent cells in its tissue context. And we also want then, or what we do is model this process in organotypic skin models, in which we can introduce cells at different stages of the cellular senescence process into a context of non-senescent or juvenile cells. And now I will hand over to Christopher, who will introduce to you how we integrate some of these modalities using tissue cytometry approach on the TissueFAXS and the StrataQuest systems.
Thank you, Florian, for the kind introduction into the topic. And I'm continuing now with our workflow and the results. So we use neural network nuclear segmentation in combination with user-guided AI classification to detect the epidermis in our sections. And in the contour of the epidermis, we measure the nuclear density, and by this distinguish between the basal layer, which is nuclear rich, and the stratum corneum, which lacks nuclei. And from there on, we continue to subdivide it further into the first differentiating layer and the rest of differentiated higher suprabasal cells. And around each nuclei, we generate the measuring mask and an additional nuclear ring mask to measure stainings at the border of the nuclei. So we export a huge variety of measurements from these masks, and we plan to use a lot of them for future clustering and prediction experiments. But at the moment, let's focus on the morphological markers. especially the size or the area and the compactness of the nuclei. And in our data set, we have 4 donors per age group with roughly 20,000 cells per donor. So first finding when we checked the distribution of cells inside the epidermis was that there's a shift of relative proportions from the higher suprabasal, so the high differentiated cells, to the more higher proportion of basal cells in the age donor, which is due to reduced thickness of epidermis and thus change of the relative distributions. So digging a bit deeper into the data, we found that there is a differentiation dependent and age dependent increase of nuclear size. So the higher differentiated the cells are, the larger the nuclei get, and also the aged samples showed increased nuclear size. And same was true for the compactness. So the basal cells that are more squished together, they have a less round, less compact nuclei. And with differentiation, we see a trend towards rounder nuclei, and the aged cells or cells inside the aged tissue also showed higher grades of compactness compared to the young ones. And additionally, we measured lamine B1 staining intensity in the nuclear ring mask. And lamine B1 is a member of the nuclear envelope, and the loss of lamine B1, as Flo mentioned before, is a well-established marker for aging. And as we expected, there's a decrease of staining intensity in the aged tissue. So to conclude this first part, we saw that there's a shift of proportions from higher suprabasal towards basal layer due to epidermal thinning in aged tissue. There's an age-dependent increase of nuclear size, and we see also an age and differentiation-dependent increase of nuclear compactness, and as expected, an age-dependent loss of perinuclear lamine B1 staining. So to determine the enzymatic activity, we use a Formersan-based enzymatic activity assay that employs the tissue native dehydrogenases and reflects different metabolic pathways depending on which enzyme you utilize. And previously, we were able to show that upon UVB irradiation, there's an increase of G6PD activity, and we were able to link peak activities to cells positive for the DNA damage marker gamma H2AX, highlighting again that G6PD provides the reducing equivalents and DNA building blocks for DNA damage repair. And now we went on to investigate the activity of G6PD and for other dehydrogenases in young and aged tissues. And these enzymes serve as direct indicators for pathways in the primary carbohydrate metabolism, including the glycolysis, the oxidative pentose phosphate pathway, and the TCA cycle. So when investigating the activity of G6PD, we increase of enzyme activity with differentiation, but also an age-related decrease of G6PD activity. On the other hand, when we looked at the activity of LDH, lactate dehydrogenase, we saw an age-dependent increase of activity. In the next step, we set out to model these age-related changes in vitro And for this purpose, we adapted our three-dimensional full-thickness epidermal organotypic skin model. I know that's quite a mouthful. So in short, these are skinnies. And they consist of a collagen support matrix, on top of which we seed primary keratinocytes. And after 7 days of differentiation, they form a fully stratified epidermis. And the beauty of this reconstructed model is that we can fine-tune it to our specific research questions. In this case, we introduced a fraction, 5% of replicative senescent keratinocytes that were fluorescently labeled to be able to visualize them in the harvested sections. So before they went into the skin equivalent, our replicative senescent keratinocytes showed a change in morphology, as well as a change in the established senescence markers. So there was a reduction of CDK1 and lamin B1 mRNA expression with an increase of P16. And in the tissue sections, we were able to track them due to the red fluorescent label. So here you can see that when we introduced 5% of the senescent cells into the model system, it had a global effect on the measured G6PD activity. So these 5% cells led to a reduction of the overall G6PD activity in all strata, while we did not see the hoped increase of lactate dehydrogenase activity. But when we start comparing only the senescent cells to their neighboring control cells, there was no change in the levels of G6PD, at least no statistically significant change, while the activity of lactate dehydrogenase was highly elevated in these replicative senescent cells. So this could indicate that the increase of LDH is directly linked to the senescence processes within the cells, while the global reduction of G6PD activity may be propagated by some sort of bystander effect of the senescent cells. And with this, I want to summarize our metabolic findings. So in the human epidermis, we saw a differentiation-dependent increase of G6PD activity and an age-dependent decrease, while we saw an age-dependent increase of lactate dehydrogenase activity. And in our aged skin models, which contained 5% replicative senescent keratinocytes, the presence of these few cells was sufficient to decrease G6PD activity globally. And while there was no change of activity between the replicative senescent cells and their neighboring cells visible, we saw an increase of LDH activity exclusively in the replicative senescent cells themselves, but not in the total tissue. And with this, I want to thank the group that helped us do this great work. Rupert Ecker and Robert from TissueFAXS, with helping us setting up all the analysis profiles, and our colleagues at the Department of Laboratory Medicine and the Technical University, and you for your patience. Thank you.